Hi friends, my name is Nandita. I am from Andhra Pradesh.
First, let us discuss about meristematic tissue. Meristematic tissue is a simple plant tissue in which growth in plants is largely restricted to specialized regions of active cell division. A meristem is a localized region in which actual cell division occurs. Growth in plants is largely restricted to specialized regions of active cell division called meristem. Plants have different kinds of meristems. The meristems which occurs at the tip of roots and shoot and produce primary tissues are called apical meristem. Root apical meristem occupies the tip of a root, while the shoot apical meristem occupies the distantmost region of the stem axis. During the formation of leaves and elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in axils of leaves and are capable of forming a branch or a flower. The meristem which occurs between mature tissue is known as intercaler meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristem and intercaler meristems are primary meristem because they appear early in life of a plant and contribute to the formation of primary plant body. The meristem that occurs in the mature regions of roots and shoots of many plants, particularly those that produce woody axis and appear later than primary meristem, is called the secondary or lateral meristem. They are cylindrical meristem, fascicular vascular cambium, interfascicular cambium and croc cambium are the examples of lateral meristem. These are responsible for producing the secondary tissues following divisions of cells in both primary as well as secondary meristem. The newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or matured cells and constitute the permanent tissue. During the formation of the primary plant body, specific regions of the apical meristem produced dermal tissues, ground tissue, and vascular tissue. Now let us discuss the types of meristematic tissues or meristem. The meristematic tissues is divided on three types. First one is based on location and second one is based on origin, third one is based on function. The meristematic tissues is divided on the basis of the location are apical or terminal meristem located on tips of roots and stem. Second is cambium or lateral meristem, it is located under the bark. Third one is intercaler meristem, it is located in the leaves and internodes. Now next we will discuss about based on the origin, how meristematic tissues are divided. Based on the origin, the meristematic tissues are divided into promeristem, it's originated from embryo, primary meristem, it originated from promeristem, secondary meristem, it's originated from permanent tissues. Now let us see based on the function how meristematic tissues can be divided. Based on this, the meristematic tissue divided into protoderm, it protects from mechanical shocks, and next one is procambium, it gives rise to the xylem and phloem, and next one, third one is ground meristem, forms the cortex, pericycle and pith of the plant. Now let us discuss the classification of meristematic tissue. First let us discuss in detail of the origin and development of meristematic tissue. On the basis of origin and development, meristem can be divided into three types. Already we discussed that promeristem, primary and secondary meristem. The promeristem embryonic are also called as embryonic meristem. This meristem develops in beginning during embryonic stage. Promeristem are usually found in root apex or shoot apex. They develop primary meristem. Come to the primary meristem. Primary meristem cells develop from promeristem and also known as primary meristem. These cells are always in division phase and form primary permanent tissue. Secondary meristem. These are the meristem developed from primary permanent tissues. They are not present in the embryonic stage of the plant. Some of the cells of primary permanent tissue become meristematic and constitute secondary meristem. By the activity of secondary meristem, secondary growth takes place. Let us discuss about the based on location means position in plant body. On the base of position, meristematic tissues are divided again into three types. They are epical, intercalary and lateral meristem. Let us discuss about epical meristem. The meristem which occurs at the tip of roots and shoots and produced primary permanent tissues. They are responsible for increasing in the length of plant organs and primary growth. Examples like root apex and shoot apex. Epical meristem has region embryonic stage that are promeristem and primordial meristem, which develops in embryonic stage, and eumeristem, and which is formed from the division of primordial meristem. According to Haberlandt, eumeristem is divided into three regions, that is protoderm or dermatogen. It is the protoderm or dermatogen is the outermost layer of eumeristem. By the cavity of protoderm, epidermal tissue system is formed. It includes epidermis, root hair, stem hair, etc. Procambium or pellirome. These are long and give rise to vascular tissues. It includes xylem and phloem. Ground meristem or peribellum. The cells of this region are large, thin-walled and isodiametric. Ground tissue system is formed by the cavity of these cells. It includes hypodermis, cortex, endodermis, pericycle, pith race and pit. Now let us discuss about intercalary meristem. The intercalary meristem occurs between mature tissues and it is separated region of apical meristem. It is found in between the plant organs. By the cavity of this meristem, length of the plant organ increases. They are present in some stems of plants. They may be present either at the base of internode, for example grasses, bamboo and eucalyptus, etc. At the base of node, they are also present at the base of leaves like pinus. By the activity of meristem, length of leaves increases. These meristems are short-lived. On losing their power of division, they merge with permanent tissues. Lateral meristem. Lateral meristem occurs in lateral side of plant organ or parallel to the longitudinal axis of plant organ. Activity of lateral meristem increases the width of plant organ, so it is responsible for secondary growth. Lateral meristem are both primary and secondary in origin, means smoothly secondary in origin. There are two examples of primary lateral meristem. One is marginal meristem. It occurs at the margin of leaf. Its activity increases the width of the leaf. So total growth of a leaf is called intercalary margin growth. Intrafascicular cambium or fascicular cambium. This cambium occurs inside the vascular bundles except intrafascicular cambium. All cambia are secondary in origin and lateral meristems are cylindrical. Now, by this division, a plate-like structure is formed. Formation of leaf plate takes place by the activity of this meristem. Mass meristem divided in all possible planes and result in an increasing in the volume of plant body. Example, the formation of embryo and endosperms takes place by this kind of meristem. Now let us discuss the position of apical meristem in different plants. Apical meristem is absent in lower algae and fungi. All the cells of these plants are divisible, so they do not show apical growth. Thus, such type of growth in this plant is called diffused growth. Diffused growth also occurs in animals. Apical meristem in higher algae, example fucus, dichota, sargassum, biophytes and petridophytes, consists of a single cell. This cell is known as apical cell. This apical cell is pyramid in shape and divided into two lateral planes. Apical meristem in ferns, gymnosperms and angiosperms of many cells. Several theories have been available in relation to structure and growth habitat of apical meristem. Types of theories of apical meristems are Histogen theory, Tunica Corpus theory, mental core theory, Newman's theory or Corpus Kappa theory, etc. Now let us discuss the apical cell theory. This theory was proposed by Carl Nageli and Hofmeister and supported by Wolff. According to Nageli and Hofmeister, the apical meristem is composed of a single apical cell. This view is only applicable on biophytes and some petridophytes and some higher algae. Histogen theory. This was proposed by Hanstein in 1870. According to him, the root and shoot apex are distinguished into three meristematic regions, three layers of histone cells. These are dermatozone, peribulum, plerome. Dermatogen. This is the outer single layer of cell. These cells form uniseriate epidermis by anticlinal divisions. Peribulum. This region is situated just below the dermatogen. It forms cortex like hypodermis, general cortex and endodermis. Plerome. This is the innermost region. Steel formation takes place by division of the cells. It means formation of pericycle, vascular bundles, pith rays or medullary rays and pith takes place. This theory is only true for root apex. It is not applicable for shoot apex of higher plants because in most of gymnosperms and angiosperms, shoot apex does not have distinct difference of these three layers. Except the above described three histogens, a fourth type of histogen is present in monocotyledons. This is known as clyptrogen. Root cap is produced by clyptrogen in monocots. Root cap is produced by dermatogen in dicotyledons. One histone present in ranunculus, two histones exception. This is only occur in casuarina due to the presence of root cap position of root apex is subterminal, so maximum growth in root takes place behind the apex. In hydrophytes, a root cap is absent. Generally, root cap is single layer, but in screw pine, root cap is multi-layer. Root cap contain more amount of Golgi body which secretes mucilage which make the root slimy. A group of inactive cells present between the dermatogen and clyptrogen is called quiescent center. These cells contain less amount of DNA and synthesis of protein is also less. Now let us discuss the theory of Tunica Corpus. This theory was proposed by Schmidt in 1924. This theory is applicable on shoot epics. It is based on planes of division. According to this theory, two types of layers are found in shoot epics. One is tunica and second one is corpus. Tunica. This is the peripheral layer. Epidermis is formed by this layer. In tunica cells, anticlinal division takes place only in one plane. The anticlinal division occurs at right angles to the longitudinal axis of the cell. Surface area of the plant organs increases due to the anticlinal divisions. When divisions occurs in single anticlinar plane, they do not increase the number of layers. Generally, tunica represents only a single layer, but sometimes it is multi-layered when the outermost layers form the epidermis and remaining layers form rest type of the tissue system with the association of corpus. Corpus. The mass of cells present below the tunica is called corpus. The cells of corpus usually larger than the cells of tunica. It forms the rest of the tissue system.